Okay. Hello, everyone. My name is Yurong. I'm a PhD student at Curtin University from Australia. My topic today is tar reforming using biochar as a catalyst. So today, my talk will cover the following key points. First of all, I'll give you a brief introduction of the background of my project. That is the significance of tar removal from gasification product gas, and why we chose biochar over other catalysts. Next, I'll show you some of my experimental results and followed by some key findings and conclusions. So, first of all, why we need to remove tar from gasification product gas. So we know that gasification is an effective technology that converts solid fuel to syngas, which can be further used for electricity generation and the production of chemicals and liquid fuels. However, the raw product gas from gasification usually contains a certain amount of tar, which determines the further application of the syngas. So we need to remove tar for particular applications. Second of all, the existence of tar inside the gasifier can easily condense inside the system, causing the plugging and the corrosion of the systems. So tar removal has been considered as a key issue that limiting the commercialization of gasification technology. So far, the key technologies for tar removal can be generally divided into physical treatment using like filter or scrubber, and thermochemical conversion of tar, cracking or reforming tar at high temperatures in the presence or absence of catalysts. Among all these methods, the catalytic reforming of tar is considered as the most promising method due to its high efficiency. And also during the catalytic reforming of tar process, the tar can be converted into some useful gases like hydrogen and CO. So why using char? Because char has a number of advantages over other catalysts like nickel-based catalysts, which is expensive, and the natural catalysts like dolomite and zeolite, they can be easily deactivated with the process continues. So char is a low-cost catalyst which has a fair good performance in tar removal. It is generated inside the gasifier or a pyrolysis plant. And the deactivated biochar can be directly burned or gasified. So the disposal of the catalyst is not a problem at all. And for the gasification chars, they used to be considered as waste. So the use of gasification char as catalyst can in return promote the economic benefits of the pyrolysis plant. So my work is use some gasification biochar or like some activated biochar as catalysts to do some tar reforming experiments. So I put biochar inside the reactor and activate it in situ under steam or CO2 for a certain time. And then I feed some bio oil to generate some tar. Tar will pass through the catalyst bed and interact with the catalyst. The biochar sample and bio oil sample I used is produced from our demonstration plant. All the experiments were did at the same experimental conditions at 800. And then we evaluated the performance of the biochar in tar reforming as well as the effects of the pre-treatment, the activation of the biochar on its performance. So as expected, when you compare the tar yield with when you do not use biochar as catalyst, when you use biochar as catalyst, the tar yield decreased. And when you use the activated biochar, the tar yields reduced further. And the same situation with the UV Florence intensities of the tar samples. Which means in the tar samples obtained after reforming with the activated biochars, there contains much less content of the aromatic ring systems. So these results has showed the activated biochar has very good performance in tar reforming, and the activation of biochar in steam has clearly increased its catalytic activities. We know catalysts can be deactivated with the reaction time prolonged. So it's the same situation with biochar. When you look at the Florence intensity with the increasing reforming time, it's increased, so indicating the biochar become less active and become deactivated. This could be caused due to the coke formation on biochar, and as well as the structural changes of the biochar in the process. So we analyzed the structural changes of biochar. So our Raman data showed that during steam activation, the oxygen-containing groups increased, but during reforming, these oxygen-containing groups decreased. So it's reasonable to believe oxygen-containing groups played an important role in enhancing its catalytic activity for tar reforming. So like with the production of the oxygen-containing groups, the catalytic activity increased, which caused the decreased tar yield and the decreased UV Florence intensities. But during reforming, the oxygen-containing groups were consumed, so the catalytic activity is reduced, which leads to the increased Florence intensities. So to further find out which type of oxygen-containing groups is particularly important, we analyzed our samples using XPS. So very interestingly, the CO single bonds showed very similar trends with the total oxygen-containing groups changing trends. So we believe the CO single bonds is key groups that is responsible for enhancing the catalytic activity of the biochar. So this part of work is published if you want to have a look at the details. So the next part, I'll show you some results between the different catalytic activities between the steam activated biochar and the CO2 activated biochar for tar reforming. So as expected, the CO2 activated biochar also has better performance, has better reforming efficiency than the non-activated biochar. And the oxygen-containing trends has further confirmed that the oxygen-containing is important for enhancing the catalytic activity of biochar. So both during steam activation or CO2 activation, some oxygen were added to the biochar surface. But during tar reforming, these oxygen groups were consumed, causing the deactivation of the biochar. So are there any difference between the CO2 activated biochar and the steam activated biochar? Yes. The steam activated biochar, for the same time of activation, the steam activated biochar has a higher reforming efficiency than the CO2 activated biochar. So the similar trends is observed during dry reforming as well, which means the steam activated biochar has higher catalytic activity than the CO2 activated biochar in both steam reforming and dry reforming. There's another interesting thing is that uh, the reforming agents, the CO2 or the steam, also affect the reforming efficiency. So for both of the biochar activated in steam or CO2, uh, the efficiency during steam reforming is always higher than that in CO2. So we compared the oxygen groups and the CO groups of the fresh char and the spent char. So the uh, steam activated biochar has a higher content of the oxygen containing groups and the CO groups than the CO2 activated biochar. And during steam reforming, the reduction of the oxygen containing groups is always slower than, in steam reforming than in dry reforming. So finally, we got some conclusions. So basically, we did experiments using activated biochar on the steam or CO2, and then used as catalyst for tar reforming from the vapors generated from the pyrolysis of bio-oil. So we found that both of the activation in steam and CO2 can increase the catalytic performance of the biochar, and the oxygen containing groups that generated during activation is very important for enhancing the catalytic activity of biochar. And the steam activated biochar has a higher catalytic activity than the CO2 activated biochar, which could be attributed to the higher contents of oxygen containing groups, the CO groups. So that's my talk today. Thank you for your attention.